Alright, so now we are going to talk about leukocytes and platelets. So remember, a leukocyte is commonly known as a white blood cell, or the WBC. It is a major component of the body's defenses against disease. So when we talk about white blood cells, what we're mainly talking about are defense, protecting the body from diseases. Leukocytes protect the body against invading microorganisms and body cells with mutated DNA. And they clean up debris as well. So they're kind of always rooting around, cleaning up the body, checking on things that are in the body to see if they're invading microorganisms, and checking on our body cells, making sure that any body cells that have any mutated DNA, for example, are destroyed. Platelets are essential for the repair of blood vessels when damage to them has occurred. So they also provide growth factors for healing and repair. So let's talk about leukocytes first, or white blood cells. The characteristics of leukocytes. Although leukocytes and erythrocytes both originate from hematopoietic stem cells in the bone marrow, sorry about that one, in the bone marrow, they are very different from each other in many significant ways. So leukocytes are far less numerous than erythrocytes. So typically they're only 5,000 to 10,000 per microliter. They are also larger than erythrocytes and are the only formed elements that are complete cells. So they possess a nucleus and organelles. Remember that erythrocytes are not. They exude, they excrete, they get rid of all of the organelles, or most of the organelles, leaving more room for picking up iron, right? Leaving more room for all of those proteins and the heme groups. Sorry, I wasn't thinking groups. The heme groups to pick up the iron. So the platelets then, remember, are kind of chunks of cells, and we're going to talk more about that in detail, but platelets are little pieces, little chunks of cells. Erythrocytes have kind of gotten rid of all other organelles, so the leukocytes are the only ones that have a nucleus, and their organelles are complete cells of the formed elements. And although there is just one type of erythrocyte, there are many types of leukocytes. So most of these types have shorter lifespans than that of erythrocytes. That's another difference there. Some as short as a few hours, or even a few minutes in the case of an acute infection. One of the most distinctive characteristics right here of leukocytes is their movement. So something that's really unique about white blood cells is their movement. So remember, erythrocytes spend their days within the blood vessels, so circulating throughout the heart and the blood vessels. Leukocytes leave the bloodstream, commonly or routinely they say, leave the bloodstream and perform their defensive functions in the body's tissues. So for leukocytes, the vascular network is simply a highway that they travel and then they soon exit to reach their true destination. So they use the bloodstream to really go through there, like it says, a highway. It's very easy for those white blood cells to kind of shoot through the bloodstream using the flow of blood, and then they get to where they want to go, and then they work their way through to get to their true destination. When they arrive, they are often given distinct names, such as macrophage or microglia, depending on their function. As shown in the figure here, they leave the capillaries. We'll see that in just a moment. Remember, the capillaries are the smallest blood vessels or other small vessels through a process known as emigration. So emigration or diapedesis. So here, emigration from the word removal, or diapedesis, dia meaning through, and pedan to leap. So leaping through, diapedesis, on which they squeeze through adjacent cells in the blood vessel wall. So they are going to get to where they want to go. Some of the small areas, they leave the little tiny capillaries, the smallest blood vessels, or other small areas, and they emigrate, they leave, or diapedesis, they walk through, leap through, they squeeze through these little tiny cells in the blood vessel wall. Once they have exited the capillaries, some leukocytes will take up fixed position in lymphatic tissue, fixed positions in lymphatic tissue, in bone marrow, the spleen, the thymus, or other organs. Others will move about through the tissue spaces, so kind of like amoebas. They're just continuously expanding, and extending their plasma membranes, kind of lobbing some of their plasma membrane out there, and then filling in to move around, kind of filling it in and walking basically through, wandering freely sometimes, and sometimes moving directly towards something in which they're drawn by chemical signals. So we have some of them are in fixed positions, some of them are moving around. And those ones that are moving around can either be wandering freely, just checking it out, seeing what's going on. Others are going directly to something based on chemical signals. This attracting of leukocytes occurs because of positive chemotaxis, so movement in response to chemicals. Positive, and then chemo, like chemicals, taxis, movement. So movement in response to chemicals. A phenomena in which injured or infected cells and nearby leukocytes emit the equivalent of a chemical 911 call. So injured cells or infected cells are going to be sending out this signal like help, 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 help. And then that's the positive chemotaxis. The leukocytes, the white blood cells, are going to pick up on that and start walking that way. Walking being a relative term, but kind of meandering their way through using their little plasma membrane. As well as nearby leukocytes, nearby white blood cells will also say, hey, we need some help, and then they'll head that direction and attract more leukocytes. So in clinical medicine, the differential counts of the types of percentages of leukocytes present are often key indicators in making a diagnosis and selecting treatment. So based on the type of white blood cells that are around, like the different percentages, the numbers, the amounts that are around can kind of give an indicator of what kind of disease is happening. So for just a minute, we're going to skip over this image and we're going to talk about it first. Well, actually, I guess here we have our emigration. All right, here we go. We'll talk about this. So one, we have our leukocytes in the blood. So here you can see, and we'll talk a moment about the different type of leukocytes. We have an eosinophil, which is a type, a monocyte, and a neutrophil. These are white blood cells, and they are responding to chemical attractants released by pathogens and chemical signals from nearby injured cells. So here we have our injured and infected cells are secreting these chemical signals into the bloodstream. So you can see all of the secretions because we have some pathogens here. So we have a cut, for example. We have pathogens that have gotten into the cut, and these nearby cells are saying, help, help, help. And so we have our white blood cells there. And then in 2, you can see these white blood cells are going to squeeze between the cells. Let me scroll down just a tad here. So here are all of our chemical help signals. And then you can see the white blood cells are going between these cells. Here are our little tiny epithelial type cells here in the blood vessels. And then you can see they're squeezing in between these cells, getting out into these other cells here in the skin in this example. So they're squeezing between the cells of the capillary wall as they follow these chemical signals to where they are most concentrated. So this is the positive chemotaxis. So they are emigrating to the site of injury and infection. And then number 3, I'll scroll up a little bit more. Within the damaged tissue, the monocytes differentiate into macrophages. So here within the damaged tissue, they've gotten to where they're going. The monocytes differentiate into macrophages that phagocytize the pathogens. So here, turned into a macrophage, and it's going to gobble this guy up. The eosinophils and neutrophils are going to release chemicals that break apart the pathogens. So you can see here, this is the eosinophil, is releasing cytotoxic chemicals from their granules into the tissue. So they're kind of exploding these chemicals out that then you can see here are bursting open our pathogen. They are also capable of phagocytosis. So we have our macrophages, which are mostly our phagocytosis cells, which we'll talk about in a moment. But others are also capable of phagocytosis. All right, so let's talk about our classification of leukocytes here. So when scientists first began to observe our stained blood slides, it quickly became evident that leukocytes could be divided up into two groups. And the two groups are divided up according to whether their cytoplasm contained these visible granules. So did it look spotted or did it not, is how they basically split them up. So let's take a look at that. So we have our granular leukocytes, as one group is called. Granular leukocytes. They contain abundant granules, pretty easy to remember, within the cytoplasm. You can see that down here, and we'll talk about them in a little bit more detail, but you can see these dots. These are our granules in each of these. They include neutrophils, eosinophils, and basophils, and you should know which ones are granular leukocytes and which ones are agranular, and that's the other group, leukocytes. Okay, and what makes them granular, which you can see here, which are the little granules or little tiny dots within the cytoplasm. While granules are not totally lacking in agranular leukocytes, remember A is without granules, agranular leukocytes, they are far fewer and less obvious. So agranular leukocytes include monocytes, which mature into macrophages that are phagocytic, and lymphocytes, which arise from the lymphoid stem line. So granular leukocytes, remember, neutrophils, eosinophils, and basophils. Agranular leukocytes are monocytes and lymphocytes. So let's take a closer look at our granular leukocytes. We will consider granular leukocytes in order from most common to least common. So all of them are produced in the red bone marrow, and they all have a short lifespan. So we'll look at different lifespans of other ones here, but these three, the granular leukocytes, have a short lifespan of hours to days. They typically have lobed nuclei. They have a lobed nucleus and are classified according to which type of stain best highlights their granules. So that was how they were named, based on the different types of staining that really brought out the granules. All right, so, and actually we can just talk about that right here for a moment, and then we'll get into more detail. But a neutrophil has small granules. You can see they're smaller than some others that stain light lilac, and a nucleus with 2 to 5 lobes. So this big purple lump here, this is the nucleus here that's lobed. An eosinophil granules are slightly larger and stain reddish orange, and its nucleus has 2 to 3 lobes. A basophil has large granules that stain dark blue to purple, and it has a 2-lobed nucleus, 1, 2-ish. The most common of all the leukocytes are neutrophils. So we have lots and lots of neutrophils. Normally comprise 50 to 70% of total leukocyte count. So over half, about half to over half of all of the white blood cells are neutrophils. They're about 10 to 12 micrometers or micrometers in diameter, significantly larger than erythrocytes. So remember, white blood cells are much larger. They're called neutrophils because their granules show up most clearly with stains that are chemically neutral. So neutrophils are stained by neutral chemicals. All right, let me scroll up here. The granules are numerous, as we mentioned, but quite fine. Up here, light lilac, as we saw. The nucleus has a distinct lobed appearance, so our neutrophils have highly lobed nuclei, may have 2 to 5 lobes, so many lobes, very clearly lobed. It increases with the age of the cell, so it seems that in general, the more lobes, the older the cell. So older neutrophils that have increasing numbers of lobes are often referred to as polymorphonuclear. So poly, many, morpho, changing, morph, and nuclear, changing nucleus. So many changed into nucleus. So a nucleus with many forms, or simply polys. The younger ones the younger and immature neutrophils begin to develop lobes and are known as bands. So we have our polys and our bands, polymorphonuclear neutrophils and bands. All right, so that's a little bit about the description of neutrophils. What do they do? So neutrophils are rapid responders to the site of infection. So they quickly, very quickly, remember there are lots of them, they very quickly get to the site of infection. They are efficient phagocytes with preference for bacteria. So if we scroll back up here in our example, remember up here we have our neutrophil. And then, whoa, sorry about that. We have our neutrophil, and then, let me see if I can fix this. All right, just talk about our neutrophil here. And then remember it said that others, right, not just our middle guy here, which was the monocyte that changed into the macrophage, but we also said that the others are also capable of phagocytosis. Sorry about that. All right, so rapid responders to the site of infection. They're efficient phagocytes with a preference for bacteria. So our neutrophils get to a site of infection, and then they prefer bacteria. So we see these a lot for bacteria. Their granules include lysozyme, which is an enzyme capable of lysing or breaking down bacterial cell walls. So neutrophils, their granules, remember those little dots, are filled with lysozyme. And lysozyme, remember enzyme, it's an enzyme that lyses, lysozyme, or breaks down bacterial cell walls. Also oxidants such as hydrogen peroxide and defensins. So that also you can remember defensins as part of the defense system here of neutrophils. They are proteins that bind to and puncture bacterial and fungal plasma membranes so that the cell contents leak out. So when I think of a neutrophil, I think of it getting that message saying, okay, there's an infection, we have a bacterial infection, we need to go right now and get that and take care of it. So they're rapid responders. They go really fast to the site of infection, and what they do is they then release those little granules. They break open, they release their lysozyme into the environment. The enzymes then are just going to go through and they're going to break down those cell walls, so start shooting holes in the bacteria. I kind of picture it as these little guys that just kind of shoot out enzymes, that when the enzymes hit the bacteria cell walls, it just eats away at the cell walls, and then it kind of breaks apart into pieces and releases all the bacterial internal stuff into the environment, so it's no longer a viable living bacteria that's causing infection. It also releases hydrogen peroxide, which is an oxidant which can kill cells. And then also I think of it as shooting out these little proteins, these defensins. It's their defensin proteins that then get stuck on bacteria walls and then just shoot little holes through them. You know, they bind to the wall and then puncture it, stick holes in the bacteria wall, so that then the bacteria shrivels up, loses all of its contents, and then dies. As well as a fungus, a fungal plasma membrane. So it sticks onto a fungal plasma membrane, starts poking holes in it, so that all the contents leak out. Abnormally high counts of neutrophils can indicate infection. Remember the different types of white blood cells, if it's higher or lower, can indicate specific diagnosis. So high counts of neutrophils indicate infection or inflammation. Particularly, remember this one is a bacteria. It's likes bacteria, preference for bacteria, but are also found in burn patients and others experiencing unusual stress. So a way that our body tries to handle stress, it's like, oh, we're stressed out, there's stress on the body, we need to send out our white blood cells to go make sure everything's okay and try to take care of stuff. And then also with burn patients, a burn injury increases the proliferation of neutrophils in order to fight off infection that can result from the destruction of the barrier of the skin. So the body says, oh my goodness, we don't have skin on that part of our body. That means that we could get a bacteria infection anytime. So it's important that we have plenty of neutrophils going through the bloodstream and going around to make sure that if an infection does start, it gets caught right away. It says we don't have this barrier that protects us from bacteria, so we need to increase our production of neutrophils in response to that so that we're prepared if the bacteria starts to infect us. Low counts then may be caused by drug toxicity and other disorders, and may increase an individual's susceptibility to infection. So there are particular drugs and pharmaceuticals that and other disorders that may make it to where you're susceptible to infection, or a person is susceptible to infection because they're not producing the neutrophils. We're decreasing neutrophils due to these things. And then bacteria, since it has a preference for bacteria, if there's bacterial infection, it may be a problem. All right. So those are the neutrophils. Remember that those are the most numerous in the body. Next we have the eosinophils, which typically represent 2 to 4% of the white blood cell count. So we have our neutrophils, which is a huge amount, 50% to 70%, and then we get to the next, the one that has the most, the next most, is eosinophils, which are only 2 to 4%. They're also 10 to 12 micrometers in diameter. The granules of eosinophils stain best with an acidic stain known as eosin, hence the name eosinophils. The nucleus of the eosinophil will typically have 2 to 3 lobes, and the granules will have a red to orange color. The granules of the eosinophils include antihistamine molecules, which counteract the activities of histamines, which are inflammatory chemicals produced by basophils and mast cells. So, the granules of eosinophils are antihistamine molecules, anti meaning against histamine, so they counteract histamine. And then histamines are inflammatory chemicals that are produced by basophils and mast cells. So some eosinophil granules contain molecules that are toxic to parasitic worms. If a parasitic worm enters the body through the integument, or when a person consumes raw or undercooked fish or meat, and there is a parasitic worm, then we would see an increase in eosinophils because they're able to release their granules, release that toxin, and then it's going to be toxic to the parasitic worms and take care of the parasitic worms. Eosinophils are also capable of phagocytosis and are effective when antibodies bind to the target and form an antigen-antibody complex. So when there is an antibody bound to the target saying, this is a bad guy here, let's get rid of it, it's forming an antigen-antibody complex, then an eosinophil will come, pick up on that flag, the antibody flag, saying, hey, come over here, I got a bad guy, and then it can phagocytize it. So high counts of eosinophils are typical of patients experiencing allergies, right? So if we have an inflammatory chemical, what that means, right, is that if a particular body is allergic to something, let's say a particular pollen, there's pollen in the environment. When that pollen molecule gets into the body says, this is not self. It says, this is not us, this is a danger. In response to the this is a danger, the body starts to make these inflammatory chemicals saying, okay, what we need to do is we need to block this. We need to somehow get this out of our body. So it makes these inflammatory chemicals produced by the basophils and mast cells, which we haven't talked about yet. So we have the basophils and the mast cells saying, sending their messages out there saying, there's an attack, there's something dangerous. Okay. And then what we have are antihistamine molecules that are going to calm that down. So we have the attack, the histamine attack, saying, let's do various things depending on the situation. So let's make more mucus in the nose so that we catch more of the pollens. Let's make the eyes water to try to flush out any of the pollen that's getting in the eyes. All these types of things that we think of as allergic reactions. And then once we feel like the body feels like it has flushed these things out and the problem is gone, the pollen is gone, then we have antihistamine molecules that are going to calm down that inflammatory response. So we see that eosinophils are going to be releasing their antihistamine molecules to counteract the activities of the histamines, which was in response to something that the body thought was a danger. But then we have to get back to homeostasis. And then they'll also release molecules that are toxic to parasitic worms. And then they're also capable of phagocytosis, particularly with the antibody-antigen complex. All right, so high counts of allergies, that's what got us on that topic right there, the general allergies response. Parasitic worms, right? If we have parasitic worms, we're going to be seeing more eosinophils to produce that toxin. And some autoimmune diseases. So that antibody-antigen complex being formed saying, hey, this is not self. Then the antibodies say, hey, look, I found a cell that is not self. And that is seen in autoimmune diseases, which we'll talk more about when we talk about immunity. Low counts may be due to drug toxicity and stress. All right. So basophils are the next granular leukocyte we're going to talk about, and the least common of the leukocytes. So comprising less than 1% of the total leukocyte count. Smaller than neutrophils and eosinophils, just slightly, 8 to 10 micrometers in diameter. And then these granules stain best with a basic or alkaline stain. So basophil for the basic stain. Basophils have large granules, remember, that pick up that dark blue stain and are so common that they may make it difficult to see the nucleus, actually, because they're so large and they stain so solidly. So in general, basophils intensify the inflammatory response. They share this trait with the mast cells, which we'll talk about momentarily. So inflammatory response. In the past, actually, mast cells were considered to be basophils that left circulation, but now they're considered to be from two different lineages. So the granules of the basophil, or basophils, release histamines, which contribute to inflammation, and heparin, which opposes blood clotting. So high counts of basophils are associated with allergies, right? So histamines contribute to inflammation. Again, we have that response to an allergy. We say we have to go through this process. Let's protect ourselves. Part of that is inflammation. So we see that associated with allergies. Parasitic infections. If we have a parasite in, then we will release histamines to contribute to inflammation. The area will be inflamed. And hypothyroidism, so a decrease, hypo meaning low, thyroidism, so low release of the thyroid hormones, or decrease function of the thyroid. Low counts are associated with pregnancy, stress, and hyperthyroidism. All right, so those are our 3 granular leukocytes. Now we're going to talk about the agranular leukocytes. So A meaning without, and then granular, granules, leukocytes. They contain smaller, less visible granules, right? So A meaning without, but we do know that they do have granules. They're just much smaller and less visible. The nucleus is simple in shape, sometimes with an indentation, but without distinct lobes. So our granular leukocytes have the lobed nuclei. Our agranular leukocytes do not have distinct lobes, but they could look kind of bent, so sometimes it can be a bit confusing. But the granules are really what give away the granular versus agranular. There are 2 major types of agranulocytes. Those are the lymphocytes and the monocytes. So first we'll talk about lymphocytes. They're the only formed element in blood that arises from lymphoid stem cells, right? So the others arise from the red bone marrow. Although they form initially in the bone marrow, much of their development is in the lymphatic tissues. So they develop and they reproduce in the lymphatic tissues. They start out way back in the bone marrow, but then most of their life and development and reproduction is in the lymphatic tissues. Hence the name lymphocytes, in the lymph tissues. They are the second most common type of leukocyte. 20 to 30% of all leukocytes. I said earlier, sorry, let me just make sure I back up for a minute. When I said that the neutrophils, remember the neutrophils we saw, where is that? The most, right here. Most common are neutrophils. And then I said after that, we have the eosinophils, which make up a very dismal 2 to 4%. I was thinking and talking in that at that point at the granular leukocytes, not all the leukocytes as a whole, right? So we look up here and we have the neutrophils, which are 50 to 70%. And then we have down here the lymphocytes, which then are 20 to 30%. So that's out of all of the leukocytes. And then it happens to be out of the granular, or agranular rather, leukocytes, the one that we have most of out of the two. All right. So they are essential for the immune response. You can imagine if they're in the lymph, that's immune, they're necessary for the immune response. The size range of the lymphocytes is quite extensive. Some even categorize it into 2 different size classes, and others 3. So there are large cells at 10 to 14 micrometers. They have a smaller nucleus to cytoplasm ratio and more granules. The smaller cells are typically 6 to 9 micrometers with larger volume of nucleus to cytoplasm, creating a halo effect. A few cells may fall outside these ranges at the 14 to 17 micrometers. So this is the 3 size range classification where that comes from. So the 3 major groups of lymphocytes include natural killer cells, B cells, and T cells. So we're talking about there are agranular leukocytes. In the agranular leukocyte category, there are lymphocytes and monocytes. When we talk about lymphocytes, we then talk about different types of lymphocytes. We have the natural killer cells, the B cells, and the T cells. So the natural killer cells, the NK cells, are capable of recognizing cells that do not express self-proteins on their plasma membrane. So all of our cells have these proteins on the plasma membrane, and all of the body's cells that are supposed to be here have these self-proteins on their plasma membrane. Those that are not self, so bacteria, other pathogens, will have other proteins, and our body recognizes those as not self. So the natural killer cells, or NK cells, recognize cells that do not express the self proteins on their plasma membrane, or that contain foreign or abnormal markers. So these non-self cells can include cancer cells, cells infected with a virus, and other cells with atypical surface proteins. Thus, they provide generalized non-specific immunity. So they're not very specific. They just generally go through, and anything that is not self, that does not have self proteins, they're going to go in and take care of. They're going to get rid of. So the larger lymphocytes are typically these natural killer cells. B cells and T cells, then, are also called B lymphocytes and T lymphocytes. They play roles in defending the body against specific pathogens. So again, we're talking about lymphocytes as a whole. We have natural killer cells, which are non-specific immunity, meaning anything that's not self, it's going to try to take care of. Then B cells and T cells have specific immunity, and they have specific pathogens that they are involved with. They're involved in specific immunity. So one form of B cells, the plasma cells, produce antibodies or immunoglobulins that bind to specific foreign or abnormal components of plasma membranes. So this is referred to as humoral immunity. All right, so it's binding to very specific components of a plasma membrane. This is humoral immunity, or body fluid, because these are floating around in the body fluid, in our tissues, in our blood. T cells provide cellular level immunity by physically attacking foreign or diseased cells. So these are actually attacking cells, T cells. A memory cell is a variety of both B and T cells that forms after exposure to a pathogen and mounts rapid responses upon subsequent exposures. So the memory cell is a type of B cell or a type of T cell that is formed after a B cell or a T cell has identified something that is not self, and then it makes these memory cells to say, hey, let's remember this particular pathogen, so that when this particular pathogen comes back into the body, if it does, then we can attack it faster and get rid of it faster. So unlike other leukocytes, then these memory cells live for many, many years. This is our immunity. So when we're exposed to something, our body produces these cells so it can attack it faster next time. B cells undergo maturation process in the bone marrow, bone marrow. T cells mature in the thymus, T cell thymus. This site of the maturation process gives rise to the B cells and T cells. So B in the bone, T in the thymus. The functions of the lymphocytes are complex, and they're going to be covered in the lymphatic and immunity system chapters. So we'll really get into some detail on those. The smaller lymphocytes are the B or the T cells, but it's really hard to differentiate those, and we won't need to differentiate those. So abnormally high lymphocyte counts are characteristic of viral infections, as well as some types of cancer. So we see them in the virus because they're very specific. They're going to bind to it, take care of the virus, and then say, hey, we need to produce these memory cells to remember this virus so we can attack it better, faster, more efficiently next time. Abnormally low lymphocyte counts are characteristic of prolonged illness or chronic illness and immunosuppression, including things like HIV infection, drug therapies, things that involve steroids. And then the other type of A granular leukocyte are the monocytes. So they originate from myeloid stem cells. They normally represent 2 to 8% of the total leukocyte count. They're easily recognized because they are very large, 12 to 20 micrometers, and they have indented or horseshoe-shaped nuclei. Macrophages are monocytes that have left circulation, and then they phagocytize debris, for example. So they go out, they leave the circulation. We saw that in the image earlier. They leave through between the two cells in the blood vessels, and then they go out there and they phagocytize things like debris, foreign pathogens, worn out erythrocytes, and other dead, worn out cells or damaged cells. So macrophages also release antimicrobial defensins or chemotactic chemicals that attract other leukocytes to the area. So they will get out of the bloodstream, they'll find an infected area, and then they will send out those chemicals. They'll say, hey, over here, and then that's what other macrophages are going to go to. They're going to be attracted to that to go and help with the infection. Some macrophages occupy fixed locations, again, and others wander through the tissue fluid. So abnormally high counts of monocytes are associated with viral or fungal infections. They're going to go out there and try to take care of the virus, cells that are infected with a virus, as well as fungal cells, tuberculosis, and some forms of leukemia and other chronic diseases. Low counts are caused by suppression of the bone marrow. So if there is an issue with the bone marrow, then we're not going to be producing monocytes that are coming from these myeloid stem cells in the red bone marrow, and then we won't have a high amount of the monocytes circulating through the bloodstream. All right, so now that we've talked about the different types of leukocytes and what they do, now we can talk about the life cycle of leukocytes. So most leukocytes have a relatively short lifespan. We talked about that already generally, typically measured in hours or days. But remember, there are those that are not that short. Remember, our memory cells that we just spoke about here last longer than that, right? Our B cells and T cells that are memory cells, that they're a little bit higher up there, right here. Memory cell is a variety of both the B cell and T cell that sticks around for much longer, for years, just in case there's another attack of something. So, but typically, all of the other leukocytes have our shorter lifespan of hours or days. They all start in the bone marrow under the influence of CSFs and interleukins, which we spoke about. Secondary production and maturation of the lymphocytes occurs in specific regions of lymphatic tissue known as germinal centers. So the secondary production, so producing more, and maturation, so their maturing process. In the lymph tissue, the germinal centers, so germinating, that growing, maturing in the germinal centers in lymph tissue. They're fully capable of mitosis and may produce clones of cells with identical properties. So this allows them, excuse me, allows the individual to maintain immunity throughout life to many threats that have been encountered in the past. So if we think about things like, say like chickenpox, for example, we have our leukocytes that were sent out to try to help with chickenpox. They were attacking, but at the same time they were making those memory cells, the B cells and the T cells that are memory cells. And then since those memory cells are able to divide, are capable of mitosis, then they can produce clones of those memory cells that were once made in response to exposure to chickenpox. And then that allows immunity throughout the life of the organism, throughout the life of the human. So now we're going to take a look at disorders of leukocytes. So the first one is leukopenia. It's where there are too few leukocytes produced. So leukopenia, or penia, is when there are too few leukocytes. And if this condition is pronounced, so if it is really pronounced, I guess is the correct term here. So if this is severe, severe case of it, there are too few leukocytes, and the individual may be unable to ward off disease. So even something, a very, very common, let's say allergen is in the environment, then that could cause a very serious situation for a body, or a small cut gets a little bit of bacteria that in a normal person would just cause a little bit of inflammation, a little bit of infection, and goes away in a couple days, may be unable to ward off that disease, and it could spread and become a serious issue. Excessive leukocyte proliferation is known as leukocytosis. So leukopenia is too few leukocytes. Too many or excessive proliferation is known as leukocytosis. Cytosis, cyto, cell, many cells, production of a lot of leukocytes. Although leukocyte counts are high, the cells themselves are often non-functional. So that's why it is a condition. There are too many. So when the body is going through the process based on various things where they're making too many leukocytes, oftentimes they're not functional at all. So even though the count may be high, they have leukocytosis, they are not functional. So then this also leaves an individual at increased risk for disease, because even though they have lots and lots of white blood cells, if they're not functional, then they can't do what they're supposed to do. Leukemia is a cancer involving an abundance of leukocytes. So in leukemia, if you've heard of leukemia, the cancer before, it is an overproduction of leukocytes. It may involve only one specific type of leukocyte from either the myeloid line, so myelocytic anemia, or the lymphoid line, so lymphocytic leukemia. In chronic leukemia, mature leukocytes accumulate and fail to die. So in chronic, if a person has leukemia for a very long time, then these leukocytes are going to be produced, an abundance of leukocytes, and then they're going to accumulate, they're going to stay in the body, and then they fail to die. In acute leukemia, so in a short-term, not a lifelong leukemia, there's an overproduction of young, immature leukocytes. So in both conditions, the cells do not function properly. So just like in leukocytosis, even though there are an abundance of white blood cells, and you'd think, well, that's fantastic, right? The body will just do a super great job of fighting off any infections. It'll take care of that really fast. That's not often the case. All of these white blood cells are produced, and they just don't function. So then they just become a hindrance. There are lots of them, particularly in the chronic leukemia. There are lots of them. They fail to die, and then they become a problem because it's just excess cells that are not functional. Lymphoma is also a form of cancer in which masses of malignant T and or B lymphocytes collect in lymph nodes. So lymphoma is where the T and B, or T and B, lymphocytes collect in the lymph nodes, the spleen, the liver, and other areas. So we have this collection of cells. I guess I could probably bring this down just a tad here. So lymphoma here is another type of cancer. So we talked about leukemia, now we have lymphoma, and then we have these masses of cells that are collecting in these areas. And the same with leukemia, these malignant leukocytes do not function properly, so they're not adding to the health of the individual. And in that case, they're vulnerable to infection. Some forms of lymphoma progress slowly, so it tends to not be too much of an issue. It can progress slowly, and then it can respond to treatment. Others can progress really fast, and you can see lots of masses of these malignant cells throughout the lymph system, causing serious problems. It comes on quickly and requires aggressive treatment, without which they're rapidly fatal. So it can become a serious problem, a fatal problem if it's not treated, and sometimes it's also just too aggressive in that case. All right, so now we're going to take a look at our third formed element. Right, we've looked at erythrocytes very in detail, we've looked at leukocytes in detail, and now we're going to look at platelets. So you may occasionally see platelets referred to as thrombocytes. Okay, so they can be called thrombocytes, but remember, cytes is an indicator of a mature cell. However, platelets are not mature cells. So here it mentions, because the name is it suggests that they're a cell. It's not actually accurate. So a platelet is not a cell, but it's a fragment of the cytoplasm of a cell called a megakaryocyte that is surrounded by a plasma membrane. So there's the plasma membrane and then cytoplasm inside of it, and it's a fragment of it, a piece that's come off of a megakaryocyte. Megakaryocytes come from the myeloid stem cells. They are large, typically 50 to 100 micrometers in diameter. They have an enlarged lobed nucleus. As noted earlier, we talked about thrombopoietin, which is a glycoprotein that's secreted by the kidneys and the liver. It stimulates the proliferation or the production of megakaryocytes. Remember, blast is an immature type of cell, blast, that then matures into the megakaryocytes. Remember, cytes is a more mature cell. These remain within the bone marrow tissue, and then they ultimately form this platelet precursor extension. So they kind of have these extensions that come off, which are plasma membrane, that then are cytoplasm filling up these cytoplasmic plasma membrane extensions. And then they extend through the walls of the bone marrow capillaries. So remember, the capillaries, we have that thin layer of cells. Between those, the walls of the bone marrow capillaries, we have these extensions, and then they release into the circulation thousands of cytoplasmic fragments, each enclosed by a plasma membrane. These enclosed fragments then are what we call platelets. That's why they're not actually cells. Thrombocytes indicate cell. They're not actually cells because they're just chunks of cytoplasm surrounded by plasma membrane. Each megakaryocyte releases 2,000 to 3,000 platelets during its lifespan. So following the platelet release, the megakaryocyte remnants, which are little more than cell nucleus, are consumed by macrophages. So after it's kind of done its thing, released all these platelets, the remaining tiny nucleus with a little bit of other stuff around it is then gobbled up by macrophages and then recycled. Platelets are pretty small, 2 to 4 micrometers in diameter, but numerous. So we typically have 150,000 to 160,000 per microliter of blood. After they enter circulation, approximately 1/3 migrate to the spleen for storage for later release in response to any rupture in a blood vessel. So after they're made, a third of them, 33%, a third of them are going to go to the spleen just for storage, just in the case that there's been a rupture, and then they can quickly be released to go and help that blood vessel. And then they become activated and perform their primary function, which is to limit blood loss. So platelets' function is to limit blood loss. They remain only about 10 days, then are phagocytized by macrophages. So they hang out in the spleen. If after 10 days they weren't needed, there is no rupture in a blood vessel, then they're just recycled. They're phagocytized by macrophages, and that stuff's used over. Platelets are critical to hemostasis, which is the stoppage of blood flow following damage to a vessel. And we're going to talk about that in much more detail in just a little bit here. They also secrete a variety of growth factors that are essential for growth and repair of tissue. Particularly connective tissue. So they do secrete some growth factors to help repair tissue. Infusions of concentrated platelets are now being used in some therapies to stimulate healing because of this growth and repair aspect. So we'll take a look at disorders of platelets here really quickly. There is thrombocytosis. Remember, thrombocytes are a name for platelets. So thrombocytosis is a condition in which there are too many platelets. So this is a serious problem because it may trigger a formation of unwanted blood clots. So if we have these platelets that are going around, their function is to create blood clots, to be part of that clotting process. So if there are too many in the body, then we would have formation of unwanted blood clots. They'd just be sticking to each other. And so thrombosis is a potentially fatal disorder. So these clots floating around. If there's an insufficient number of platelets, that's called thrombocytopenia, the blood may not clot properly, and so then excessive bleeding can result. So if we don't have those platelets floating around that are going to be useful in creating that platelet plug, which we'll look at in a moment, then we can have some serious issues there. So really quickly here, the platelets are derived from cells called megakaryocytes. So we have our myeloid stem cell, which then produces a megakaryoblast, which then matures into a megakaryocyte. And then we have these extensions, you can see, platelet precursor extensions that reach out and then kind of break off and become our platelets that are floating around then. All right, so that is the end of talking about each of those specific formed elements. And then the next section is hemostasis. So we're going to talk about platelets a little bit more and what hemostasis is.